All right, if you didn't get enough of me in lab, here we go for some more notes on classification. That allow us to organize organisms and have a common reference point to speak with each other about what type of organism we're talking about. The main levels of classification we'll be looking at in here will be domains, kingdoms, and then there will be phylum, class, order, family, genus, species, and strain. And as I mentioned in lab, the most important levels for us are going to be the domains, kingdoms, genus, species, and strain. That is where I want you to put your emphasis. There will be 3 domains in our class, 6 kingdoms. And I say for our class because classification can be controversial, but I have found really simplifying it to these 3 domains and 6 kingdoms seems to make the most sense to me. So we'll go ahead and outline the characteristics of how an organism gets into each domain, go through the kingdom characteristics, and then we'll discuss genus, species, and strain a little bit later on. Starting with the 3 domains. First domain is going to be the domain Archaea. This one was named because of the ancient, very old, archaic, ancient types of cells. They are extremely extremophilic, meaning they like conditions like boiling hot springs or deep sea vents. Very extreme conditions that a lot of organisms can't withstand. A lot of times, because they are in these extreme conditions, we don't typically see them as human pathogens. However, there has been a study out of UCSF that did correlate severe periodontal disease with people that had higher levels of archaeans found in their oral cavity. So possibly there are some considered pathogens that could cause disease. It's just these are one of the hardest to study organisms. So we don't have a lot of information on them yet. The next large group will be the domain bacteria. So you are familiar with this domain. And the third domain is going to be the domain Eukarya. These are all cells. Every single cell can be categorized into one of these domains. We did just discuss the characteristics of prokaryotic cells. So I will say prokaryotes. And I think you can tell by the name of this domain that this is going to be a eukaryotic organism. We are not going to spend a huge amount of time on the archaeans. Just remember they are archaic, ancient, and extreme in their environment and nutrition. However, there will be a couple of characteristics that I will want you to compare and contrast, so I'll try to go straight across with those characteristics. A few things that cells all have in common would be that cell membrane. So they all have a cell membrane. Cell membrane, and cell membrane. And they all have DNA. However, this is that single circular chromosome. DNA, single chromosome. And this one also has DNA, multiple linear chromosomes. All right. Now, just with that information, do you already know what part of that cell looks like and its replication strategy? Yes. So since there are multiple linear chromosomes, that is going to require a much more compartmentalized cell structure. And that compartment the DNA is going to be found in will be the nucleus. We did say the nucleus will divide via mitosis. And an average epithelial cell in good conditions, we could estimate dividing in about 20 hours, compared to the nucleoid of these cells will be dividing via binary fission. And we'll say, for bacteria's sake, the fastest bacteria in good conditions could possibly multiply every 20 minutes. That is extremely rare, but it can happen. Cell membrane, DNA. What else do cells have? Ribosomes. And that was sort of a unique find with the domain archaea, that they actually have a combination of 70S and 80S ribosomes, usually free-floating, versus the domain bacteria. Of course, you know, those are all 70S ribosomes. Then we get to the domain eukarya, and here you have your 80S ribosomes. The other characteristic all cells have will be cytoplasm, but I'm not going to write that down. You'll remember that. Okay, now let's just transition down into that kingdom level. Even though I don't have it lined up perfectly, I'm just going to bring these down. We'll have the kingdom archaea. Everything that's written about the archaeans up here holds true for that kingdom as well. So there is really no difference in the kingdom or domain archaea. Some people do not differentiate between the kingdom and domain archaea. Same is true for the kingdom bacteria. Every now and then you will hear the kingdom Monera, that would be sort of the old school, or eubacteria, true bacteria. However, we're trying to simplify here. I'm just going to keep it simple with the domain bacteria enclosing that kingdom bacteria. Same is true for the domain archaea. I'll have a couple of lines down here to try to divide these visually. If there are 6 kingdoms, as I noted on the left, then if we have 2 here, the domain eukarya is going to have 4. In the 3rd kingdom, we will find the kingdom Animalia. That's where we fall. Then we'll have the kingdom Plantae. I'm just going to abbreviate K, Plantae. Kingdom Fungi. If it doesn't fit into one of those 5, it will fall into kingdom Protista. Ooh, am I running out of room here? Yes, I am. Well, darn it. I'm going to move the video camera over. Okay, back over so you can see those protistans. How does an organism get into each of these kingdoms? Well, for these domain archaea cells, these all have to be single cells. Will be single cells. These will all be single cells. These all have to be multi-celled organisms. So animals in their mature state will be multi-celled. Yes, we do start out as a single cell, but as a mature organism, they are multicellular. The same is true of plants. They are also multicellular. Fungi will have both. They have single or multi. The yeast will be an example of a single-celled fungus. The molds will be an example of a multi-celled fungus. Then to get into the kingdom Protista must be a single cell. Okay, moving on. That's one differentiating factor between these. Another differentiating factor is going to be the presence of a cell wall or not. And for the archaeans, they do have a cell wall. And we are just going to refer to that as a special cell wall. You do not need to know the ingredients or composition of that archaean cell wall. Yes, it's special. It allows those organisms to survive in very harsh extreme conditions. Complicated chemical name, we're not going to memorize that one. The cell wall composition that you will be memorizing are going to be found in the domain bacteria and the domain eukarya, starting with the kingdom bacteria. Their cell wall is going to be made out of a very important hybrid molecule that we call peptidoglycan. And we'll see this is almost like a chain link fence type of structure, very strong, very rigid, allows organisms to survive in sometimes challenging environments. The animal cells, however, like us, one thing to remember is there is no cell wall. So no cell wall for us. Cell wall, none. An experiment that we do would be RBCs or red blood cells in water. They will lyse because that cell membrane is not strong enough. Cell membrane not strong enough for osmotic pressure. So the cell wall is a very strong component of cells, and it can be a benefit if cells do have them. The kingdom Plantae, you will have a cell wall in plants, and the composition is going to be cellulose, carbohydrate you can identify with that OSE ending, kind of like sucrose, fructose. The kingdom fungi, they also have a cell wall. I'm kind of losing my linearity. The composition is going to be chitin, which is another carbohydrate commonly found in the exoskeletons of crustaceans and insects. It gives you an idea that that is a relatively strong type of molecule. When we get to the kingdom Protista, the cell wall varies. Cell wall varies And it's going to vary between none, that would be your animal-like protistin, or it may be your plant-like protistin. Can you see that? I think so. Enough. Whoops. Just put that down a little bit. Okay. Looking at there, you might notice the cell wall actually is a good differentiating characteristic between these organisms. If I told you or said I'm imagining a cell and it's composed of chitin, automatically you could tell me what type of cell I am talking about, or at least identify what kingdom that cell falls into. So a cell that had chitin, and chitin is a carbohydrate found in the cell wall, would be found in that kingdom fungi. You're not sure if I'm talking about a yeast or a mold until I state I'm looking at a single-celled organism that has a cell wall of chitin. That would be enough information to then lead you to identify yeast. So comparing and contrasting, remember when we are looking for unique attributes, those can be good clues to help us identify what type of organism we're looking at. Another thing I'm going to mention is back to this peptidoglycan. If I said penicillin is a chemical and it is designed to injure or prevent the synthesis of peptidoglycan. That is its target. It will bind to the enzyme that produces peptidoglycan. Since it is very specific in targeting peptidoglycan, do you think it's going to be able to target any of these other kingdoms? No, because that peptidoglycan is specific for bacterial cells. This is one of the reasons that antibiotics like penicillin will only target bacteria, and they will not have an impact on those fungal infections. Peptidoglycan is not found in that kingdom. When we think of viruses, did I mention that viruses have a cell wall? No, because they don't. And there will not be any peptidoglycan found in viruses either. That is the reason penicillin is not going to be effective at all against a viral infection. It's not worth giving a patient any extra drug if it is not going to help them, because no drug is really healthy for us. All right, these will be the major characteristics to remember. Again, be prepared to compare and contrast all of these. We'll walk through some of the questions in the PowerPoints that I have posted online.